our final speaker this evening, or is it evening already? I'm sorry. This afternoon, this morning, is Lisa Cleckner. And Dr. Lisa B. Cleckner is the director of the Finger Lakes Institute at Hobart and William Smith Colleges in Geneva, New York. She holds a PhD in environmental health sciences and an MBA. She enjoys collaborative efforts that engage different constituencies to include faculty, research scientists, federal agencies, community organizations, and outreach groups. At Finger Lakes Institute, Dr. Cleckner has developed initiatives addressing invasive species, harmful algae blooms, mercury in fish, and watershed management across the Finger Lakes. Dr. Cleckner's talk today will address what has been learned about the biology and ecology of phytoplankton from a scientific buoy in Owasco Lake. And let's welcome Dr. Cleckner. Are you okay?
Again, no joke on the brightness. So it's my pleasure to be here today. Thank you for having the Finger Lakes Institute present, following on John Halfman's legacy, and certainly appreciate all of the work that he has provided. And I know that, there's literally some big shoes to fill. So just want to provide some updates on some of the things that we are doing, particularly in relationship to the buoy and relating it to harmful algal blooms. So general overview of where we're going today. I'm going to give a little bit of an introduction because this is my first time speaking, but then also present some data from the buoy from 2023, talk about HABs, what's the connection there, and I'm going to finish up with data portals, which is one of the things that we have been working on and have received some funding from Cayuga County to basically have some Owasco Lake focused data and some graphics online. And certainly want to always acknowledge the Emerson Foundation for a lot of the investments they've made at Hobart and William Smith Colleges over the years. We are always greatly appreciative of those funding. So since it is my first time, much like Kirsten did, I'm just going to give a very brief overview of who I am and some of the things I've worked on. My father was in the Air Force, so I'm a military brat and ended up in upstate New York during my high school days. And from there, it was like, wow, there's a lot of great institutions, and I went to Colgate University majoring in biology, and to Julie's point, my watershed moment, my senior year, was taking a freshwater biology class, a freshwater algal ecology class. And I was like, wow, algae, who knew, right? And so, I've been looking at algae for a number of years and knew that I wanted to pursue water things moving ahead to graduate school. So this gives an opportunity. I've been using this composite slide. I went to graduate school at the University of Michigan, huge Michigan football fan, and I'm sure everyone knows the 2024 national championship. Don't get a chance to say it often, so I'm going to take my moment. But yes, so I did my PhD work on Lake Michigan and worked on the EPA research vessel and did a work with trace metals and mercury. And that led to my postdoc. I was at the University of Wisconsin in Madison. I did a lot of mercury research looking at trophic accumulation of mercury. So starting at lower trophic levels, again, starting with that freshwater algae and going up through fish and understanding how the pollutants would build up. So the Everglades had a chance to go work with alligators. I mainly liked the smaller fish and the shrimp, but still it was fun to see alligators. And also did work in Lake Superior, including Isle Royale, and having a chance to do some work there was amazing. And then I think I just took a step back and I'm like, okay, I need to start doing work as I'm getting older where maybe I can be the top of the food web instead of being at the will of maybe some other predators. So with that, I decided to return back to New York, and I was like, oh, New York has lakes. It's a beautiful place, family in the Finger Lakes. And so I just kept coming back to visit, and I was like, wow, it's really beautiful here. And ended up going to the Simon School in Rochester to get my MBA, and from there I worked at Syracuse University for a number of years working on applied research and environmental, basically collaborations, trying to commercialize technologies in the space of indoor environmental quality, clean and renewable energy, and water quality. Really great opportunity to learn the landscape, and started at the Finger Lakes Institute in 2011. So just a little bit of background, and I know that people have, certainly know John from, he played a big part in establishing it, but we have now been around for 20 years, and I think some of the key things we're trying to do is actionable scientific analysis. Obviously, at an academic institution, a lot of professional development for our students and community education. And we recently revamped our mission. And really what we're about is taking those findings, the translational research part of it, taking those findings and putting them into collaborative action. And we do that in a lot of ways. We do actual projects ourselves, but we also try to write a lot of grant applications. So if we think of invasive species and where we get money from, the government is a big part of that. So being able to find funding and go after funding to address things like watercraft stewards out at the boat launches, or to do hydrilla treatments in Cayuga Lake. We've been writing a lot of those proposals in collaboration with many other things. And I just want to take a moment. I know this is the Bob Brower Symposium. I will say Bob was the impetus for a lot of the invasive species work that we do at the Institute. He was one of the first ones to reach out to me once I started. We wrote a grant together. Of course, our first grant was not funded, but it led to other conversations. And that's how we were able to get funding to start our watercraft steward program across the state. So really, I'm indebted to Bob and just want to acknowledge that. And so it's a cool moment. And so, within the Institute itself, we have not only research, but we have a K through 12 education program. We do a lot of outreach as well. We realize that's really important. Our research conference is just one example. And then watershed stewardship, again, the word of the day, I believe it is. So I will say from over the years of doing different ecological research, all of these issues are related. When we think of land use and nutrients, we need nutrients in our water to basically drive our, the growth of organisms in the water. Food webs are very important, but that balance is obviously really important. Invasive species can certainly come into systems. They can change the cycling of nutrients. They can, change the quality of habitats and things like that. And then contaminants are all impacted by this as well. So some of our mercury research that we published in the 2010, basically in 2019, is showing the impact of things like the round goby, or we can think of quagga mussels, how they have basically impacted food webs for fish and where the fish are found. And so we tend to look at these as very interrelated issues, and working very much on the applied side of the house, trying to look at new watershed management practices and adapting to things like climate change and trying to anticipate what's going to happen in the future. So one of the last things I'll just mention very briefly about the Institute is we do have a New York State certified an ELAP lab for nutrients, total suspended solids, and total microcystin. We've had that now for a couple of years, and that has been a great opportunity for us just to expand some of our programs. So I'm going to spend the next part of the talk basically talking about the buoy itself. If you've been on Owasco Lake, I'm sure you've seen it, but in 2023, the Finger Lakes Institute staff, we took over the operation of that, and we received funding from Cayuga County to keep that going. The buoy itself was purchased in the early 2010s. John Halfman was part of a foundation grant that was able to purchase a buoy, about $150,000 or so. And with all the work that has been done in Owasco Lake and with support of the Emerson Foundation, the decision was made to put the buoy into Owasco Lake so that we could learn more about it. We had one in Seneca Lake previous to that, starting about 2008. And so the more that we can have these buoys working in different lake systems, we learn about not only the lake that we're studying, but those comparisons are really important. And from a scientific perspective, that's really the basis of what we do. We look at what's been done in the past and look at what's happening in other systems that we can then do models. So what does it mean to actually have this buoy out there? We have the buoy, and hopefully you can see the map. Yep, okay, good. Where the point is it's in an offshore location. It's the buoy, we try to get it out in usually around April. Last year, there was a little bit of a delay due to some funding issues that we had at the beginning, but we were able to put it out anyway, and we left it out until November 1st. So that buoy is out there sitting in the lake. We'd have to do calibration checks. We go out and visit it every couple of weeks, and also retrieve the data that the buoy is collecting through cell phones, and having basically an online portal where we can display the information that it's collecting a couple of times a day. And this 2 times a day means that the buoy itself collects 2 profiles in about 150 feet of water. And those times are midnight and noon, and we take measurements at about every 1.5 meters. Just want to acknowledge Trevor Massey from our staff. He worked closely with John on a lot of the basically putting in the launch of the buoy every year and the retrieval, and it's no small enterprise to do that sort of work. So really, what is a profile? And I just wanted to do a quick graphic to show what that is. Essentially, we have this XO2 sensor, or it's a sonde, is what it's called. It's a YSI is the company that makes these. And so we have this sonde, it's on a chain, right? So if you look at the bottom of the sonde, there's different sensors there, and those sensors are tuned to measure different water quality parameters that we care about. So rather than having a scuba diver that would literally take a sonde and go, up and down or hold it, or throwing these over and doing casts every day, we have a profiler. And so as part of the buoy system, essentially it's set up with a computer to just go down and take measurements about every 1.5 meters, and then collects the data and then sends it back to a computer on campus. So why is it important to measure these profiles? Why do we care about this? Well, with lakes, what really helps us understand what's happening, especially with regard to algae, phytoplankton, things like that, is where the nutrients can be found and how the layers of the lake mix with each other. And those layers of the lake mix with each other dependent, that basically it's changes in density. And those changes in densities can be due to changes in temperature, and it could also be due to changes in dissolved materials in the water. So if you think of like salt water and fresh water, the salt water obviously has a lot more salinity in it, and it influences how fresh water and salt water is more dense. So basically, if you have fresh water and salt water, typically the salt water will sink and the fresh water floats on top. So we know that based on the sun heating the water, that essentially the surface layers start to warm up, but then once there's enough of a temperature difference between the different layers, the layers don't, can actually start to separate. So you get a warm zone, the epilimnion, or kind of the surface waters. You get a zone where the sun doesn't penetrate as deep, and you get very rapid change, and that's called usually the thermocline. And then you get the deep water where it's just cold. And so we know that the lake turns over. Most lakes turn over 2 times a year. Some of the Finger Lakes only do once a year, and it's just dependent on ice. And the last thing I'll say is water is its most dense at 4 degrees C. So that means water that is basically 4 degrees C is going to sink to the bottom, right? And if it's colder than that, then you get ice formation and it floats. So that density change is really important. So with the buoy itself, we deployed it last year on May 3rd it was, and pulled it out on November 1st. So the parameters that we measured, depth, temperature, specific conductance, blue-green algae, chlorophyll, dissolved oxygen. Obviously, the fish and organisms need dissolved oxygen in order to breathe. And for each one of those parameters, we collected almost 11,000 measurements during the time that the buoy was out. And so I want to just kind of walk you through what the data itself looks like. This is a graph of temperature with depth. So this is essentially taking a cross-section of the lake. And on the y-axis, you have depth. So the top of the graph is the surface of the water. And that's always a tricky thing to learn in Excel. It's one of the first things water people have to learn is how do you get your axis to go the other way? So that's with depth. Time is on the y-axis, on the x-axis. So you can see we started in May, went through November, and then the colors represent the measurements of the temperature over time. And so I said, we do 2 profiles a day. So, through computers, you can do this interpolation between your measurements and develop these images or pictures or visualizations of the temperature change over time. And you can see, and just kind of, I did, of course, a lot of the temperatures they're plotted in Celsius, but I do have the Fahrenheit equivalents for those. So you can see in mid-June, and this is 2023, you start to get some pretty good warming up in the surface waters. And, you kind of get those zones where that warm water tends to definitely stay on the top, and it doesn't really mix much with the deeper layers below. So you're about 77 in the red, orange areas. In the orange itself, it's about 70, 68 degrees. As you get down into the greens, you're 60, and then to the, obviously the deeper we go, hitting that 4 degrees C with the blues. So I think, if you think of this graphic, it basically represents, instead of me showing you 33,000 numbers, this picture is a representation of what those 33,000 observations are. Now, the other thing we can look at is really what the productivity of the algae are over time, and when does most of that productivity happen? And in most lakes, you really get these springtime changes that are very intense algal growth. We know phytoplankton, there's a lot of different types. If you've heard of a diatom bloom, that's typically what you're going to get in the springtime, and the chlorophyll is represented on that top left graph where you're seeing that yellowish to greenish area. That's kind of some of the most intense chlorophyll concentrations that we measured over the season. But you can also see pulses. But the other thing that's important is, you have to look at light coming in and where the nutrients are and where, basically the layers where you're going to get that growth of the phytoplankton. And it tends to stay very much at the top of the water column, relatively speaking. We're talking about the top, 10 to 15 meters. The other important part of this then is, just thinking of photosynthesis and what some of the products are. Once you get algae growth, then you get oxygen produced, and you can start to, see the dissolved oxygen concentrations, and the saturation is a way of just expressing how much there is in the water column relative to the temperature and depth. It accounts for temperature. And so those yellow areas in the bottom left panel show that there's some really intense oxygen production, and obviously those do correspond to where we're seeing chlorophyll. Now, as you kind of go through the summer and you see still, over just looking at the temperature graph that's over on the right, you can still see that the warm layers are not really mixing with the other, the layers below. And you see this kind of gradient or kind of the thermocline, and you start to see, where dissolved oxygen starts to decrease as time goes on. And it's just because particles are settling out of the water column, and using up oxygen as they're degrading. So if you think of the cycling of nutrients and in a lake, that's what happens over time. So again, these visualizations, and the buoy is really important to understand that. Another parameter, the specific conductance, this is really a test of the ionic strength of the water. And so if you think of ions, you think of things like, that have a charge, basically. So if you think of like calcium, magnesium, or, sodium and chloride. So if we do like a lot of salt applications over a particularly snowy winter, it can drive up the specific conductance of the water over time. And so this helps us understand, but you can also see very similar to the growth and the different layers. So if you have a higher specific conductance measurement, it means the water is denser, it's going to sink. But you can also, if you think of other things that are coming into the lake, like rain, it usually has a much lower specific conductance. Streams can also come in. A lot of them actually have higher specific conductance. So it's good to understand some of these density changes. And again, this helps to show just how much the materials are mixing or not mixing in the layers throughout the water column. We'll come back to this. So I want to take a moment here and just shift into a slightly different topic. Harmful algal blooms, going back to that freshwater ecology class, very important, for our region, everything that Seth mentioned. I borrowed some information from the NIHAB's website and some of the images that basically the volunteers from AULA and other partners had submitted through NIHAB's. I know people know that, are familiar with that, so I'm not going to spend a lot of time, but it does show all of the blooms across the lake for the season. And not going to do it, but there it is. It's Toledo, right? The Great Lakes, more blooms in the Finger Lakes, drinking water supplies, sun, nutrients, warmer temperatures, and little wind is kind of the prevailing paradigm. One of the things I learned when I did take my freshwater algal ecology class was how incredible cyanobacteria are. And cyanobacteria obviously are the organisms that produce HABs and can produce toxins at times. And they have they're 3.5 billion years old. I saw the title of an article is basically like, we're all just living in the cyanobacterial world. And really, they are the ones that have dominated our planet over time. They produce the oxygen that allowed us to be here. But they have these qualities that provide them with not only survival mechanisms, but competitive advantages compared to other phytoplankton. So they can move up and down in the water column. They're kind of like the Goldilocks. They want to find the best place for light and the best place for nutrients. They have not only, chlorophyll, as all, the phytoplankton do, but they also have accessory pigments that allow them to absorb energy, from a number of different wavelengths. And so these accessory pigments are called phycocyanin and phycoerythrin. And so cyanobacteria can really absorb light from across the spectrum. The morphology of them, the shape, they form colonies. They're really tough to eat for filter feeders. If you think of plankton, zooplankton and things like that typically eat diatoms and green algae. And they also have different ways to find nutrients. Some of them can actually fix nitrogen from the atmosphere. So we talk about nitrogen and phosphorus. Phosphorus is the limiting nutrient in freshwater ecosystems, but nitrogen is also a consideration that we're learning more and more about. And when all else is equal and you can grab nitrogen from the air, that's a competitive advantage. And we've also learned relatively recently, I would say, but that basically the nitrogen in the system has a lot to do with the toxins that are produced. So we need to pay attention to both phosphorus and nitrogen. I'd say the other thing, kind of when I learned all about cyanobacteria, it was always like in shallow, warm lakes. And that's what they said, well, those are the lakes that are gonna basically have HABs. And now we know that low nutrient lakes also have HABs. And so we're continuously learning more and more about this. Even Lake Superior at this point, as you know, when Ken presented the Great Lakes, I mean, it's hard to imagine that this is actually happening in Lake Superior now. So I think when we think about nutrients, and I'm not going to go into this in great detail, but these are just some of the issues that we're continually exploring. Nearshore versus offshore sources of nutrients and timing and fate, the bioavailable forms. And basically, if there's low concentrations in the water, does this mean that there's no nutrients? Because a lot of times, with water quality, we go and measure these nutrients. But a lot of times you're trying to measure something that's already been basically it's already been consumed by the organisms. So if something was there, but then it's not there, how do you actually, study it and say, well, does low nutrients really mean that the phosphorus is never there? And it's like, no, it's just that it's already gone because it's in great demand. So a lot of times we have to really focus on sufficient time resolution to look at changes due to processes. And water sampling in and of itself is important, but it is by no means the only way that we can understand this question. So I think we've been talking about HABs in the Finger Lakes for a number of years. I mean, I think 2017 was like one of the years that got everyone's attention. And science, I will say, we're patient, right? So we're starting to get papers out there at this point, and just want to show here's a couple that have come out recently. Tony, I know he's in the audience, published something on basically looking at cyanobacteria in Canandaigua Lake. And one of the questions is, are the blooms coming from nearshore areas and going offshore, or are they coming from offshore going nearshore? Because obviously wind has a big impact on this. People talk about quagga mussels and how they've altered the nutrient cycling into nearshore areas, and there's a lot more light availability, so the productivity is going up. So there's a lot of these intertwined questions. But in this paper that Tony and his co-authors published or wrote, they showed that the HABs were developing in the offshore area, and that there were signs of them being present before we were seeing them in the nearshore areas. And there's a lot of other information in there too. Tony, please correct me if I'm wrong, but it was a great contribution to helping us, especially in the Finger Lakes context, to understand what's going on. John Halfman and some of our other colleagues at Hobart and Williams colleges also did some nearshore work over a number of years, and this was published last year as well, and ties in a lot of the Buoy data. So looking at some of the times that we start to see blooms, a lot of them were triggered after these large precipitation events and mixing of, if you will, of the water column. So mixing those layers of the different parts of the lake. And then I just want to point out there was another recent paper, and I know there's colleagues here from Community Science Institute, but we were on a paper with Ruth Richardson and Nan Wang from Cornell University, and thinking about what a bloom is. The bloom is not just cyanobacteria. It also has its own microbiome. So that means, and with all of the advances in DNA and RNA work and genetic materials, we can start to understand now that, we can think about what can, basically make nitrogen or phosphorus available. And we can look at that from a chemical place. But we can also think about all the bacteria besides the cyanobacteria that are part of the aquatic system or a biome that helps to make those nutrients available. And we're learning more and more about those as we move forward. And this starts to tease out some of the things that we've seen in blooms from Cayuga Lake. So what does a HAB look like under a microscope? And hopefully when I play this short video, you can start to see these are very dynamic things. And so this big blob kind of swimming at the bottom is Dolichospermum. It's a cyanobacteria. It has some protozoans, Vorticellas, associated with it. And you can see that it's not just a single organism. There's other types of cyanobacteria like Microcystis are in it. And there's a lot of other bacteria that are part of the system. And you can see different, when well, we won't go there, but basically there's a lot of different materials in this, and bacteria are really, really important. So then kind of to start wrapping this up, so then what is the connection with the buoy? I'm just gonna do a couple takeouts here, very just short stories related to a couple of things that we might be able to relate to the buoy. But so the first is the nutrient concentrations by depth. So we know that things like seiches that are present in our lakes, and they cause undulations in these layers, so that, while it may look like, basically there's a very strong demarcation, we know that there can be swings in the layers of the lake of 5 meters, 5 to 10 meters, even on Owasco Lake. So I stole a paper or a figure from John from one of his presentations. So the top left is the graphic that I showed earlier. The bottom left is, basically it's a more detailed look of what's really happening with the temperature change. And you can see these up and down movements across the thermocline. It has to do with seiches. John has a paper that he just came out with co-authors from NOAA and some other organizations, looking at the physics of this. And I'm gonna say, biologist here, I'm fascinated by the numbers, and I'm starting to really appreciate this. But there's a lot of math that you can work out to actually predict these, and some of the changes related to when they get disturbed or not. So then, since we were doing the buoy work this year, we can't help ourselves as scientists. Like when we go out and sample, we always have to do extra things because, I don't know, because we have all this time, I guess, and no money, and that's why we do it. But we're always curious, right? So what we did is we did some grab samples of nutrients, and we took some while we were out there and analyzed them in our certified lab. And so phosphorus is the top graph there, and nitrogen is the bottom graph, and the 3 different layers of the lake. I'm going to just say surface, the thermocline, where all the changes, and then the deep part. And for total phosphorus, there's no difference of the concentration in those 3 layers. The nitrogen shows a different story. And so just, food for thought and thinking about, the thermocline is notoriously hard to because of those undulations, so trying to think that you're grabbing something at about 10 meters deep, the thermocline could actually be in a different place that day. So it's a little bit of a challenge to do some of the sampling, but between the buoy and taking a look at that, it can give us some good insights as to where we would want to sample that day. The other thing that Tony looked at in his paper was kind of like this emergence of HABs, or when they begin, and started to look at an algae torch and some of the other field probes. So one of the things that we looked at from the buoy data from the 2023 set was to look at the ratio of the phycocyanin, so that is specifically for a pigment that's found only in the cyanobacteria, to chlorophyll, and tried to look at changes in that ratio over a season. And so when you start to see some lighter colors, like the yellows to the oranges to the reds, and sorry for the colorblind people here, that's the other thing that scientists, we always do, but you can start to see the areas. So like even in June, we're starting to see some interesting things with depth. And this is not the full water column. We kind of subsampled a little bit more of the surface water. But you can start to see some indications that this cyanobacteria and some of at least the ratio of the phycocyanin to chlorophyll is increasing. And on the right is all the data reports. The first HAB, I believe, was reported in June. There were about 70 events on NIHABs. They went until the end of October. So we're just looking more and more into this data analysis. The next thing I want to talk about are these whiting events on the lakes. Sometimes it can be confused as a HAB, and what these are it's a calcium carbonate particle that's formed during photosynthesis. And I'm going to just start a video, and this is from Owasco Lake. I think I'm going to start a video. We'll see. Yep. So this was out on our Boston whaler. It was in September. And there were a lot of blooms that year. You can see the kind of the yellowish, greenish water color close to where the pier is, but then you can start to see these white particles formed. And so this is a pretty astounding event. And if you've ever heard of coccolithophores and thinking of ocean cycling and ocean whiting events, you can see these things from space. And there was so much of the calcium carbonate, basically particles in the water as we were sampling that day. And I just want to remind us again, as we look at things like specific conductance and look at why the shallow waters are actually lower, this would be one of the ways that you could actually, these particles could drop out of the surface and go to the bottom eventually. And that's a way to remove this, so that's why the specific conductance is decreasing. I mean, whiting events have been published. I found this reference, 1987, so we know about these, but it's just another way to put some of the information that we're collecting into context. Last part, and I know we're getting close on time, so I will do this quickly, is we have been working on data portals. And the idea of this is to use basically public, we want people to look at data, and we want to make it easy for them to look at data. And so we've taken a lot of the work that John did over time and a lot of his nutrient data and published, it's all now available specifically for Owasco Lake on the website. And the other piece that we've added is this opportunity to make graphs of this information. So some people are numbers people, some people like pictures. I'm a picture person. I like numbers too, but I really like the visualizations. So the opportunity to actually make these graphs is available, and we've been working on curating data sets to include in this portal. We're also doing this with the Buoy data. So at this point, we have the 2023 data from the Buoy available, and maybe you want to look at a specific time frame, or you want to look at what's happening with the wind speed, the wind direction, with some of the meteorological parameters that were also collected. You can start to look at these online and to do your own customized review. And some people like this, some people don't. I think it's kind of cool. So really, these are really meant to pull together different water quality data. And we're also working on some other updates, trying to get data sets from the stream monitoring that has been, that's going on as part of the 9-element working group. And obviously ALA's efforts, and also adding things like English units to things. So some takeaways on the cyanobacteria. I mean, again, we're living in their world. They're amazing organisms. They can do a lot of different things, but mixing and upwelling events are important. Hydrodynamics, understanding storm events, and obviously not just high nutrient lakes. The other thing with the buoy itself is it really provides a frame for understanding changes in the lake over time. It's great that we have 10 years worth of information so far on temperature with depth, but as we look at climate change and some of the modeling, the stuff is going to continue to change. So having that physical information about the lake is really, really important. Just want to thank my colleagues at the Finger Lakes Institute, Invasive Species, K-12 education, some watershed managers and lab folks and data managers. Indebted to them for all of their efforts. And then finally, I just want to thank John Halfman for all of his pioneering work he did on Owasco Lake. And as we look ahead to the 2024 field season, we're going to do a lot more of phytoplankton sampling. This is on our boat in Seneca Lake. But trying to have nets that are big enough that you can send them deep enough into the water column for the times that we're trying to sample, because we know some of these organisms go up and down related to light conditions, but also to make sure that we're able to sample them appropriately is important. So with that, thank you very much.